last semester, and that was not ideal. But anyway, also I should say I've gotten a couple of questions about recordings where you can access them. You should have gotten an email with the recordings in it. If you didn't get that email, let me know. Yes, I can send the, yeah, I guess you could call it as a worksheet. I don't know why I was going to say put quotations around that. It's a little bit of a different format than what I usually do. But there is the kind of sheet that I have for tonight. It's sending in the chat, it's just taking a minute. So yeah, like I said, a little bit of, I don't want to say a little bit of everything. It's very much, tonight's session is not a comprehensive review whatsoever. I thought if we were going to try to cram 8 lectures into 1 hour, it would be super overwhelming. So I definitely recommend that you go back, look at stuff we've done at PAL sessions already this semester. Yeah, because tonight is not comprehensive. It's a lot. I think almost everything I'm talking about tonight is stuff that we didn't talk about in my regular sessions. So with that being said, starting off with Hardy-Weinberg equilibrium problems. So we did talk about Hardy-Weinberg to some extent in my regular sessions, but we kind of talked about more what it is, what it does, and like the 5 conditions that have to be met, or the 5 assumptions. Yeah, I didn't show or go over any calculations, and Amy did say today that you should be kind of familiar with basic Hardy-Weinberg problems, nothing like super intense, but the basics of it. So yeah, this can be an area that's confusing for sure, to say the least. So I thought it would be a good idea to kind of go over some of those. So yeah, the sheet. No worries. Yeah, if you ever join after I've sent the sheet and you want it, oh no, it's not sending. I don't know why it does this. If anyone knows Teams better than I do, and you know why sometimes it won't let me send twice, or send the document twice, I'd appreciate that. But okay, it says it won't let me send it. Give me 1 second. I'll send it to you in an email if that works.
Is there any way you could send it to me in an email? My chat isn't coming up.
Yes. Oh, okay. Okay. Yeah, I can definitely send it to you in an email, Emily, for sure. And some other people have sent it to the chat. So thank you. That's super helpful. Okay. So yeah, Emily, I will send it to you really quickly. Okay. And if anyone else wants it and your chat is not working for some reason, let me know. Okay. I think we're good. Okay. I think we're ready to roll. So, Hardy-Weinberg, basically what you need to know above all else, I guess, are these 2 equations and then what each thing within those equations actually means or represents. So your 2 equations are p squared plus 2pq plus q squared equals 1, and then p plus q equals 1. So they're pretty straightforward. It's open notes anyway, so you'll have access to those. But what's more important than just knowing what the formula or the equation is being able to understand what each figure, I guess, represents. Does someone have a question? Okay, maybe not. I think someone became unmuted and it was just me echoing. Okay. So, starting with, let's start with this one, the P plus Q equals 1. It's a little bit simpler, I guess. So P represents the frequency of the dominant allele in the population, and Q represents the frequency of the recessive allele in the population. So basically, to think of an example just off the top of my head, you could have, there's a question after this one. I think it's butterflies that we talked about. So let's say there's blue butterflies and red butterflies, and blue is dominant over red, just in this example. So P is going to represent how frequently the blue allele appears in the population. Q is going to represent how frequently the red allele appears in the population. So it makes sense that they add up to 1 because they're frequencies. So P could be 0.75. So 75% of the alleles in that population are blue. And then Q would be 0.25 because the other 25% are red. But you can see why it has to add up to 1 because it's dichotomous. It's one or the other. In that you have to have one of the two alleles. Or okay, you can, I shouldn't say one of the two because you can be, you know, homozygous dominant, homozygous recessive, or you can be heterozygous and have one of each. But there's no like third option here. Okay. And so then looking at this equation, P squared represents the percentage of homozygous dominant individuals. So this isn't, it's not how frequently the allele is present, it's how frequently the homozygous dominant individuals are present. So in a population of 100, maybe 40 of them are homozygous dominant, so P squared is going to be 40. Similarly, Q squared is the percentage of homozygous recessive individuals, and 2PQ is the percentage of heterozygous individuals. So that's kind of the big distinction, is that P and Q represent alleles, whereas P squared, 2PQ, and Q squared represent individuals. Okay, so that can be confusing, especially without a lot of context. So let's go through some of these practice problems. Also, copyright, I took these straight from a website online. I think it was like the University of Kansas, maybe. So I hope they don't come for me. But anyway, so starting with, you've sampled a population in which you know that the percentage of the homozygous recessive genotype is 36%. So using that 36%, calculate the following. So the first being, what is the frequency of that little a genotype? So that homozygous recessive genotype, what's the frequency? That is actually given to us in the question. So we know that the frequency of the genotype is just going to be this 36%. It's the percentage of individuals that are homozygous recessive. So 36%, or I guess they're the same thing. So I'll put them both. Okay. So it's not confusing. So we know that. That's kind of the first step. So just to go back and compare, I guess, this would be the homozygous recessive individuals. So that's going to be our Q squared. That is the percentage of the population that are homozygous recessive. Okay. So to find the frequency of that little a allele, we said that little a is recessive. So we're looking for the frequency of the recessive allele. So we're looking for Q. Little q. And we want to figure out, or obviously we want to figure out what Q is, but sorry, that I was kind of getting ahead of myself. Okay, so we said here the frequency of the little a genotype is 36%, and we said that that percentage of homozygous recessive individuals is Q squared. So we know, maybe I should have put it over here, that Q squared, whoops. Sorry. Q. No, that's not what I want. Oh my goodness. Q. There we go. Almost. So close. There we go. Okay. So we said, yeah, Q squared, which is the percentage of homozygous recessive individuals, is 0.36 or 36%. Okay. And so we're looking for the frequency of the A allele, which is just Q. So Q we can find by taking the square root of Q squared. So the square root of 0.36 is 0.6. Should have been able to do that in my head, but actually I have a good point on that. We'll get back to that later. Okay, so to get Q, all we did was take the square root of this 0.36 value. So okay, I think that's straightforward. If not, let me know. So the next thing that we want to do is find the frequency of the big A allele. So that is P. Okay, so we know we have that one equation, which is P plus Q equals 1. So we can rearrange that to say P equals 1 subtract Q. And we know what Q is because we just did it in the previous question. So nope, not Q. P equals 1 subtract 0.6, which equals 0.4. So P equals 0.4. The frequency of the dominant allele is 0.4. Okay. So the next thing is the frequencies of the genotypes of the heterozygous, well, starting, I guess, with homozygous dominant and heterozygous genotypes. Okay. So to figure this one out, we have to think back to kind of what our P squared value, our 2PQ value, and our Q squared values are. So we know Q squared, we know Q, and we know P. So with that information, mostly I guess we just need Q and P, which we've previously determined. We know that, watch me fight with this again. That went slightly better than the last time. P squared is going to be the frequency of that homozygous dominant genotype. Maybe I'll scroll back up to this little definition. So P squared is the percentage of homozygous dominant individuals, which is what we're looking for. So P squared is, we're just going to take our P value, which we just determined to be 0.4, and we're going to square it. That's going to give us 0.16, if my math is correct. We'll just double check. Haven't taken math in a while. We're good. Okay, so that's our P squared, and that's going to represent the percentage of homozygous dominant genotypes. I don't know why I keep getting my my words mixed up tonight, so sorry about that. And then to find the heterozygous, so the percentage of individuals in the population that have this heterozygous genotype, we use 2pq. So that's going to be 2 multiplied by p, which we determined to be 0.4. Oops, multiplied by q, which we determined to be 0.6. So I think that would give us 0.48 objections. I'm pretty confident in that. Yep. So 48% of the population is going to be heterozygous, 16% of the population is going to be homozygous dominant, and starting way back at the start of the question, it told us that 36% of the population is homozygous recessive. And to kind of double check our work, if we want, we can add all three of those numbers up because we know p squared plus 2pq plus q squared equals 1. So we have p squared was 0.16, 2pq is 0.48, and q squared is 0.36, and that does indeed equal 1. So we're on a roll here. We're doing well. Okay, so then the last question is asking for the frequencies of the two possible phenotypes if the dominant, or if big A is completely dominant over little a. So what does that mean exactly? So this is asking for phenotypes, not genotypes. So if big A is completely dominant over little a, that means that any time, this probably wouldn't come up, honestly. This is like, you have to use too much knowledge from the previous course. But anyway, we'll go over it just for the fun of it. So, right, if big A is completely dominant over little a, that means that anytime big A is in the genotype, it is going to be dominant and it's going to be expressed. So that means that in our homozygous dominant, obviously, it's going to display that dominant phenotype, but then also in our heterozygous genotypes will display that dominant phenotype. So what that is going to look like is we know, there's a couple of different approaches to this. One's a lot easier than the other. One way you could look at it is, okay, well, I don't really like how this question is worded, to be honest. But anyway, irrelevant. So basically what it's saying is that the only individuals that are going to express that recessive phenotype are going to be the ones that are little a. And we already know from the question that the little a genotype is expressed in 36% of the population. So 36% of the population will be, what's the proper wording for that? Will show recessive phenotype. Although I don't know that phenotypes can necessarily be recessive, but I think you know what I'm saying here. So with that, you could just say, well, if 36% are that phenotype and they can only be one phenotype or the other, then you can do some quick maths in your head and just say 100 subtract 36 gives us what? No judgment, please. 64, I think. So 64% are the other guys. They'll show that dominant phenotype. You could have also figured this out by oh, wait a second. Is my 64 right? No, that's right. Yeah, my math's okay. Oh, okay. Sorry guys, I just can't do math. So the other way you could have figured it out would be by taking these two, because those two genotypes are going to give you that dominant phenotype. So you just add the 0.48 and the 0.16, and that's going to give you 0.64 or 64%. And then with that, you could subtract from 100 and get 36% will be the recessive phenotype. Okay, does that make sense, or are there any questions or concerns about any of that? I have another question to go over, just to kind of clear the air. So I think these are the types of questions you could expect to see, potentially. I mean, there might not be any, it's hard to say, but I don't think it would be anything a whole lot more complicated than that. So this is the next question, which I think is a little bit less straightforward, might be a little bit more tricky. So it says, within a population of butterflies, the color brown, big B, is dominant over the color white, little b. And 40% of all butterflies are white. Given this simple information, calculate the following. So calculate the percentage of butterflies in the population that are heterozygous. So the key to these questions is figuring out, okay, so what do I need to calculate? And also, what information do I already know? So you're looking for the percentage of butterflies in the population that are heterozygous. So you're looking for individuals. You're not looking for alleles. You're looking for those individuals that are heterozygous. So straight away, you know, you're not looking at your p plus q equals 1. You're looking at your p squared plus 2pq plus q squared. And we said that heterozygous is represented by 2pq. So you're looking for your 2pq is going to give you the percentage of butterflies in the population that are heterozygous. But first, before you do that, you need to figure out what your p and q are. So looking at the question, the only number we're given is this 40%. It says 40% of all butterflies are white. So 40% of the butterflies are white. I literally just said that. But we know that white is recessive. So in order for those butterflies to be white, they have to have little b and little b. They have to be homozygous recessive. And so we know that 40% of the population are homozygous recessive, and that is our q squared value. Q squared represents the, Q squared represents the number or the percentage of individuals in the population that are homozygous recessive. And we know that to be 0.4 from the question. Okay, so q squared, 0.4. So from there, we can find q. We just take the square root. Now, this is where you need to be careful. This is a vicious trap. This is. Listen to what I have to say, because the square root of 0.4, despite what you might believe, is not 0.2. That may seem obvious to you. It was not to me. When I first started doing Hardy-Weinberg problems, I did that all the time. I think, you know, because square root of 4 is 2. So 0.4, oh, it must be 0.2. It's not. It's 0.632. We'll just leave it at 0.63 just for simplicity's sake, I guess. But anyway, yeah, I don't really get how percentages work sometimes. That's why I'm not a math major. But anyway, don't make that mistake. That's why when I had 0.16 earlier, I square rooted it just to make sure. So yeah, use your calculator if you're not sure. Okay, so we have our q value, 0.63. And so from our q value, we can find our p because we have our equation that is p plus q equals 1. So because p plus q equals 1, we can rearrange that so that it's p equals 1 subtract q. and that's going to give us 1 subtract 0.63, and that is going to give us 0.2737. That's better. Yep, that's correct. Okay, so we have our p-value, we have our q-value. Q is 0.63, p is 0.37, so we can find 2pq. It's going to be 2. Don't forget the 2 also. I think that's a common error, that it just gets dropped somehow. For no like particular reason, people just forget about it, I think. All right, so I'm going to use my calculator on that one. Oh, I typed the numbers in wrong. 0.37 times 0.63 times 2. That gives us 0.4662, but we'll go to 0.47 just to keep it simple. So that is our answer. 47% of the butterflies in the population are heterozygous. Let me double check the website I snagged these from just to make sure I didn't make a mistake. Yes, that is what they also said, so we're good. So yeah, that is definitely an example where things can be a little bit more tricky. It's not as straightforward. You might have to use some additional, or you might have to use the information that's given to kind of squirm around a little bit before you kind of get the information that you actually want. But keep in mind, 30 question midterm, I think. Yes, and it's designed to be written in 40 minutes. So you're getting 60, but it's designed to be written in 40. So, you know, roughly a minute per question. So I don't know if it would even get to this. You could do this in a minute, probably. But yeah, I don't know. Just keep that in mind. You're not going to see like crazy calculations, or if you were at my Monday session where I did the dissimilarity matrix and went like super far with it, you're not going to see that for sure. Like you might need to be able to understand it, but you're not going to be asked to like do all that work out, because that takes definitely more than a minute. Okay. Also, I feel like I'm losing my voice a little bit, so if I'm getting loud or difficult to understand, or not loud, quiet, or difficult to understand, let me know. Okay. Next one is, I'll try to keep this given information up. It's looking for the frequency of homozygous dominant individuals. So again, you're looking for individuals, not looking for alleles. So once again, there are a couple of ways you could go about it. We'll go about it probably the most like proper way that promotes most learning. So the frequency of homozygous dominant individuals is p squared. There we go. No, so close. There we go. So p squared is the number or the percentage of homozygous dominant individuals within the population. So p squared, and we know our p-value. I shouldn't say p-value, because that's a stats thing, but we know our value of p, I guess. So we can just take 0.37 and square that thing. And that gives us 0.1369, so let's say 0.14, just because we've been rounding to the second decimal place. So yeah, that's your frequency of homozygous dominant individuals. You could have also done it by taking, you were given q squared, you've determined 2pq, and you know that p squared plus these two numbers are going to add up to 1. So you could have just taken 1, subtract your 0.4 from your q squared, and then subtract your 0.47 from your 2pq, and you get 0.13, which close enough. We rounded, so there's going to be some variation. Okay, hopefully that is helpful. Like I said, I don't think it will get more confusing or complicated than that, just given that there's not a whole lot of time. Not to say you don't have a lot of time, I guess, but it's not designed to be a test with questions that are like taking 10 minutes each. Okay. Hope that was helpful. So the next thing I just kind of wanted to touch on, I guess, that I didn't really talk about in my regular sessions was the tree of life and parsimony. So essentially, parsimony is just the choosing the most simple, I guess, the simplest route was the most likely route, essentially. So in terms of evolution, parsimony means like if you have an individual that perhaps in the past was hairless, but now it's evolved to have hair. Most likely, there was just one evolutionary change. It went from being hairless, there was one evolutionary change where it developed hair, and it continued on. Most likely, it was not, oh, it developed to have hair, and then it lost the hair, and then it developed to have hair again, and then it lost the hair again, but it's got the hair back. You know, like that's like, I don't know, 4 or 5 evolutionary changes. So that's probably not the most likely scenario. Okay, so this is a little bit big. I'm gonna size it down. I also pulled this from the interweb. It's not original content, but gets the point across. So essentially, each tree of life that can possibly be created is considered to be a hypothesis, because you're hypothesizing about, okay, how did this tree, or not how did this tree, I guess, how did these organisms come to be? Which tree of life could have been the answer? And either of these two could have been the answer, I guess. Could show us like which species are most closely related. But if you look at them, and this is like a good example because they're all notched out really well. I think this makes a good midterm question, to be honest. Again, I don't write the midterms, or I don't like make them, so I have no say in what's on them, but I think it makes a good midterm question. Is given, 2 or 3 trees of life such as this, being asked, which is the most likely, it may or may not imply that you're to use the theory of parsimony. But anyway, if you're presented with a question like that, they won't have these little notches probably, so you have to be able to kind of distinguish. Actually, yeah, it probably would have the notches. It would at least have to tell you that things were diverging. Well, I guess anyway, irrelevant. Don't worry about me getting in over my head. Okay, so essentially, you can look at these and you can count up how many changes there were in each tree of life. So here, they developed vertebrae. So that's one change. Next, these individuals, except for sharks, developed bony skeleton. And next, all of these individuals over here developed 4 limbs. Here, all of the individuals past this point developed an amniotic egg. And then we have 2 notches here where these organisms developed hair, and these organisms developed 2 post-orbital fenestrae. I'm not sure what that is, to be honest. So in total, 6 evolutionary changes. Whereas if we're looking at this tree, we have here they developed vertebrae. Here, similar to this tree, but there is a big change in that ray fin fish have been put in with sharks. So the bony skeleton evolved. So that's at 2. But the bony skeleton also evolved up here. They have it in twice. So we have to count that twice. So that puts us at 3 changes. Here we see the appearance of 4 limbs. And then here again, we see the appearance of the amniotic egg. And then over here we see hair, and again, 2 postorbital finished. So that puts us at a total of 7 evolutionary changes. So is it more likely that this tree is accurate or this tree is accurate? This one. Because it's just simpler. It makes more sense that a bony skeleton would develop just at this one point and that the ray-finned fish would be included in that than the bony skeleton would develop here and then years later it would develop here again. Just makes more sense that it would develop back here just the one time and the ray-finned fish would be included in that. Okay. If there are any questions about that, let me know. Oh my, what's going on? Okay. Short note on cladistics, because I did skip over this, I guess, when I was making my regular weekly sessions because I thought it wasn't the most important content of the lecture, but I still think it's important. If you go on in biology, these terms are really important. I find them very difficult to keep straightforward because they sound very similar to me with all, the same suffix. And again, I think it makes a good multiple choice question. You could be given a tree and instead of, well, it could still be A, B, C, D. It might ask you, members A, B, and C are part of a blank group, but A, B, C, and D are part of a blank group. That sort of question. I don't know. I think it makes a good multiple choice question. So monophyletic groups they include the ancestral species, and all of its descendants. Okay, so this is an example of a monophyletic group. Here at this fork, we have the ancestral species. So it's the last common ancestor of all 3 of these species. And so all of the descendants of that common ancestor are included in the monophyletic group. So A, B, and C. Monophyletic is not the hardest to understand, in my opinion. It's probably the most straightforward. So mono, meaning one common ancestor, and all of its descendants that came after that. So if you were looking at the common ancestor being here, then everything here would be a monophyletic group. Yeah, okay. Paraphyletic group includes some, but not all of the descendants. So here in this example, this is a paraphyletic group. We have a common ancestor here at this number 2, but G is not being included in this group. So context of, your midterm, the question could ask species D, E, and F are all included in a group, but G is not included. What type of group is this? And the answer would be it's a paraphyletic group because they all share a common ancestor, but not all of the descendants are part of the group. Okay, and then we have a polyphyletic group, which is maybe the most confusing in my opinion. So polyphyletic groups include species from different ancestors. So yeah, I kind of misspoke, I guess, earlier when I said, well, no, scrap that. We'll ignore the last 5 seconds. Okay, so polyphyletic groups The common ancestor is not also part of the group, if that makes sense. So here, yeah, I haven't been super clear, I guess. But in the monophyletic group and the paraphyletic group, it includes this common ancestor. The common ancestor is included in the group. But in the polyphyletic groups, this is the most, or I guess the last common ancestor of these three species, and it does not get included in the group. But we can have a bit more variety, I guess, because I don't want to say that necessarily. Yeah, polyphyletic groups are a bit funky, to be honest. But basically, the giveaway with a polyphyletic group is that the last common ancestor of all of the members of the group is not included. That makes sense. Again, I don't think this is like need to know stuff. I mean, it is important because it gets kind of tossed around a lot, probably in further lectures in this course, and then if you take any other courses kind of similar to this, it's important terminology to know, but it's not the meat and potatoes, if you will.
I love you.
Okay. I think that's all I wanted to talk about with those. Again, I don't think it's the most important thing, but I think it could easily come up, and it wasn't something I addressed in my regular sessions, so I wanted to just talk about it. Okay, last thing was kind of going over gene flow and genetic drift again. Yeah, these two concepts can get confused with each other a lot, I think. Yeah, I still get them mixed up sometimes, so it happens. They just sound way too much alike, in my opinion. Genetic and gene, and then drift and flow. What's the difference? There is a difference, but they sound way too similar. Okay, so genetic drift is when we have changes in the allele frequencies of a population that are due to complete chance. So they just happen randomly. So it can happen in the event that there's some sort of, well, event, I guess, where, you know, a certain portion of the population is going to be rendered unable to survive and or reproduce. So if for some reason, whatever that reason may be, and it has to be a chance reason, it has to be random. You can't say only half the population reproduced because they were the only half of the population that were strong enough to survive. No, more like half the population were killed in a landslide or something. And it didn't matter if they were big and strong or weak and feeble. If they were in the landslide's path, they died. Didn't matter. Random chance, those are the ones that died. And so that only leaves half of the original population left. So only half of the population can reproduce. But so if that happens, only half the population reproduces, and all of those who reproduce, just by, again, random chance, happen to all have the same or similar genotypes. So maybe the ones that survived the landslide just also happened to be homozygous dominant, let's say, then the other genotypes will be lost. So if we had in our population, our theoretical little population, maybe we had some heterozygotes and some homozygous recessive individuals, but they were all killed in the landslide due to random chance. And so in the next generation, we only have the homozygous dominant alleles. So all of the next generation are all going to be homozygous dominant as well. Yeah. So that's an example of how it can reduce genetic variation, especially in small populations just because in small populations, there's a greater chance that one whole allele could get wiped out. That's a bit of a dramatic example. Usually I don't think it would happen quite that drastically. But let's say, you know, we had a few heterozygous individuals survive, maybe a couple of homozygous recessive individuals, but most of them were wiped out. Then that recessive allele is going to have a hard time being represented in the population after that because there's less of it. It was recessive to begin with, that sort of thing. Okay, so there's two kind of main ways, I guess, that genetic drift can happen. So in no particular order, one being the bottleneck effect. So the bottleneck effect, actually, I should try to bring up the slide that had the picture of the bottleneck effect. So I've been vaguely talking about why it's called the bottleneck effect. But the imagery is really nice, actually. So just give me a moment. If I can remember what lecture it's from. I think it might be lecture 4. PopGen, that makes sense. Okay, I've found it. I'm going to screen grab it. Probably could have shared my screen, actually. Might have been easier, but okay. So this is essentially why it's called the bottleneck effect, is that, so you have this original population, there is some catastrophic event of some kind that is considered to be bottlenecking, that only a few survive. And those few you can see are mostly blue, white, none of the yellow ones survived. So blue, yellow, white, that is representing our genotypes. So homozygous dominant, heterozygous, and homozygous recessive. So yeah, that's, I guess, why it's called a bottleneck effect. I think that's a good kind of imagery to help understand it. Maybe I should make it tiny. I just don't want it to be confused because it goes here, not under gene flow. Okay. So yeah, that's why it's called the bottleneck effect. And then there's also the founder effect. I don't think there's like any good, really like, okay, no, didn't think so. That's okay. So the founder effect is more along the lines of you have a population and some portion of that population splits off from the big, large group and they go off on their own and they start their own new, smaller population. So they're founding a new population. That's why it's called the founder effect. So the example I always think of with this is if we have migrating birds flying those great big, I mean, Canadian geese, if you've ever been in Canada in the fall, you've probably seen or heard them migrating. They fly in huge, huge groups. Like sometimes it blows your mind how many you see together. If you're ever out in the, this is me getting on a tangent, but I've seen upwards of like 200 geese flying together. It's crazy. But anyway, so say it's a foggy day, some of the members of that goose pack, flock's the right word, get separated from the rest of them. And it's really foggy. Surely they'd be able to hear them because geese make a lot of noise, but let's say for some reason they can't hear them, they can't see them, can't find them. Eventually they give up. So let's say there's maybe a dozen from that main central group of like 100. And so that those dozen geese, they can't find their flock anymore. So they're going to just start their own flock. And again, they could have allele frequencies that are not representative of the entire population. So maybe that whole population of 100 geese, maybe they had a lot of homozygous recessive individuals. But the 12 geese that got separated from the pack, maybe they're mostly homozygous dominant or heterozygous. So they're going to have different alleles. They're going to start their own population, and the allele frequencies in that population are going to be different from the allele frequencies in the original population. Okay. And then there's the concept of gene flow, which is the confusing one to me, in my opinion. Gene flow can be described as the movement of alleles among populations due to the movement of fertile individuals or gametes. So what does that mean in simpler terms? Essentially, alleles don't move, organisms, individuals move, kind of like COVID doesn't move, or the virus that causes COVID doesn't move. People move, and we've seen over the last year that people moving can cause a lot of spread. Anyway, unrelated. But essentially, yeah, the alleles can't spread themselves to new populations, right? They need some sort of vessel to be spread. So that could be an animal, could be, you know, a grain of pollen. That's a gamete, I guess, but, you know, it can bring new alleles into a population. So let's say, I guess the example that I've given previously with this, maybe, let's try to think of a new example. I've given that one before. I always talk about deer populations, and I'm like, okay, let's try to spice it up, make it different. And I'm thinking moose, like it's the same thing essentially. Okay, it doesn't matter. I'll just go with my classic deer example. So let's say we have two populations of deer living, you know, maybe in the same forest or adjacent forests, but they haven't in the past interacted with each other. So they are probably going to have different allele frequencies. So one group might have a lot of homozygous dominant individuals because they have a lot of that, you know, big A allele, whereas the other group might have a lot of homozygous recessive because they have a lot of those little a alleles. So let's say gene flow would essentially be moving the alleles among those two populations due to the movement of fertile individuals. So the deer decide, I'm going to go across the stream and check out this other population of deer that we've never seen before. So that would be the fertile individual moving alleles among populations. So that fertile individual, let's say they hop over the stream, over to the other deer population, and they're like, Mamma mia, there are some sexy deer here, and then they reproduce. That's going to introduce their alleles into the other population because the offspring will have alleles from the other deer population that they previously or otherwise, I guess, wouldn't have had introduced to their population. I hope that makes sense. All right, that is all that I had prepared for tonight. We've got about 5 minutes left if there are any kind of outstanding questions. If not, good luck. Don't burn yourself out. That's the thing about evening midterms exams. Don't study so hard all day that your brain is exhausted by the time you have to write. Take breaks, try to get outside for a little bit, that sort of thing. Sleep well tonight, eat well. You don't want to be writing on an empty stomach, that sort of thing. And if anyone wants a copy of what I went over this evening, just send me an email afterwards and let me know. I'll send my email to the chat. Probably most of you have it by now. Or you could probably just type in my name and find me, but